Hello, everyone. Good morning. Thank you very much for your presence here. My name is Cleidy Osorio. I am from Peru, but I am working in Brazil, in Ribeirão Preto, América do Sul, University of São Paulo. The name of my principal investigator of my lab. Well, I would like to make a brief introduction about my work, talking about human African trypanosomiasis, or sleeping sickness. This disease is a neglected tropical disease causing sleeping partners, as we can see, disrupt of the sleeping partners, as we can see here. About diagnosing, it's from serological tests to lumbar punctures, as we can see here. About treatment, it is considered drugs via intravenous and intramuscular, including drugs like melarsoprol with side effects because melarsoprol is arsenic derivated drug. And the symptoms could be involved muscle pain, fever, coma, and even death. This disease is the agent causative of this disease is the Trypanosoma brucei, a unicellular protozoa with unique flagellum. This disease is localized in the sub-Saharan Africa. Why? Because the presence of the vector ZZ fly, or just ZZ. You can see here the complex life cycle of this vector, of this parasite really, changing to adapt different environments, the ZZ midgut and the human host, mammalian bloodstream. According to these characteristics, this Trypanosoma brucei is considered a key model for biological research. This parasite is composed by two components, the flagellum and the cell body. Between these two components, principal components, there is an important zone called flagellum attachment zone, or FAZ. This FAZ is a complex of protein interacting between them to some functions, vital functions really, like morphology, motility, and survival. We can divide these attachments, flagellum attachment zone, or FAZ, in three domains: flagellum domain, intracellular domain, and cell body domain. One of these proteins in the cell body domain is the FASTN protein, the focus of my work. It's a gem protein with 2,000 kilodaltons approximately. We can see here in this microscopy image the position, the localization of the FASTN in the parasite between the flagellum and cell body. We can divide it into 5 regions that we can see here. This protein has been characterized for the first time in my lab, as we can see here in this article. FASTN protein is a key protein, very important in the cytokinesis. How? It is involved in the correct, properly cleavage formation, and the correct division, distribution of the organelles in the division cell. Using RNAi to silence this protein, we can see here in this graph allow cell growth in the absence of FASTN. In absence of FASTN, we can see incorrect cell division, which we can see here in the microscopy image, generating different phenotypes, incorrect phenotypes, as we can see here. Due to, since we have demonstrated FASTN is very important for this parasite, my main objective of this work means to know about the structure of this protein, to know more about the function of this protein, this important protein, with 2 approaches, in silico and experimental, to confirm our in silico predictions. We have performed distorted predictions of FASTN to select the region for as an initial point for our structural studies. We have selected the central region as our initial point because it has low disorder, and we considered that this region, central, just the central region, is more structured. Focusing in the central region, we have performed coiled coil predictions with Markov algorithms, and we founded this coiled coil domain, and with high likelihood of dimerization as parallel coiled coil. Using the recent Nobel Prize at AlphaFold 2, we can obtain the structure, the prediction of the structure of the central region. The central region has two globular domains flanking one coiled coil domain, as we have found in the previous prediction, with 44 nanometers of length. Taking advantage of this structure, we have identified the key amino acids composing the coiled coil domain. Positioned in the amino acids in the position A and D, like leucines, they are hydrophobics to make the interaction between the two change in the dimerization. And the position E and G, as we can see here, they are polar amino acids for support this interaction. With these promising results, in silico results, promising in silico results, we want to confirm this prediction using a heterologous system to produce recombinant proteins and make biophysical analysis. We have used performed affinity chromatography, and I present to you the side excursion purification on the central region and just its globular domain. And we can see here in blue, in the central region, two peaks. This is considered like a volume, the void volume, like molecular aggregates that we are not considered equal to our analysis. And this peak, the number one, is the sample, homogeneous sample considered for our analysis. As the globular domain is a smaller molecular size, as we can see here, right? And the molecular size of the central region is 66 kilodalton, and just its globular domain is approximately 11 kilodaltons, that we can see, we can confirm in this SDS page. In this step, we don't know if central region is a dimer. That this is the focus on my work. So we have performed Segmal's analysis to know if the central region is a dimer. We can see here in black the central region with two peaks. A void column, the molecular aggregates, is characteristic of the sample. And this peak with 244 kilodaltons corresponded to a dimer population, right? The central region. I also presenting the coiled coil domain with 65 kilodaltons corresponding to a dimer of the coiled coil domain, because the monomeric size is approximately 33 kilodaltons. And in contrast to the globular domain, with just one peak of 12 kilodaltons corresponding to a monomeric size. To know if our in silico predictions and our experimental predictions are talking the same thing, we have conducted analysis by circular dichroism of this recombinant protein, and we found that both globular domain of the central region has experimental and in silico results has the same minimal value. It means that they are consistent between them. This minimal value is approximately of 208 nanometers, that we can see here, the same minimal value, and in the case on the central region, experimental and in silico analysis present the same minimal value of 220 nanometers in this point, 202 nanometers. It's very interesting that, according to literature, this minimal value of 280 nanometers corresponding to a globular proteins. That is the case of the globular domain. And this minimal value corresponding to proteins with high alpha helices in its structure. The conclusion is, I'm sorry about the change, in silico analysis help us to identify the initial point to make the analysis about the structure. And we have selected the central region, focusing the central region. This central region had two globular domains flanking this coiled coil. Is a dimer, okay? Central is a dimer, and we can conclude that all the protein is a dimer protein. This is very important for the localization and the function of this protein. And both in silico and experimental results has consistency. And our next step is to use this central region to resolve the structure, right, with techniques like X-ray crystallography. And with this recombinant protein, because we have produced a recombinant protein, we are going to use to make a follow analysis like full down, for example, to identify these molecular partners. Why? Because we consider that fasting is like organizer within flagellum attachment zone. It is very important for the parasite. It is very important for the survival of the parasite. And we consider it in the future like a therapeutic target. Thank you very much to all the people involved in my work and these people of the Instituto de Física San Carlos, lab of Professor Richard Garrett, helped us with the biophysical studies. Thank you very much for the support, finance support with this agency. Thank you very much for all of you to be here early. Thank you.
Time for questions. Well, I have one then. Why working with Trypanosoma brucei, which is African, which is an African pest, and not with, for example, Trypanosoma cruzi, which affects Brazil and other countries?
It's a good question, really, because in my lab, the majority of people work with Trypanosoma cruzi, but in our case, we have identified this protein in this parasite, right? Important protein and interesting protein, really, because the punctation of the dimerization, and because that is a jam protein, and we found fasting in cruzi also, but it's not a jam protein, and our lab is focused more in jam protein. It's just in jam protein, and at the same time, Trypanosoma brucei not affected in America, but it is possible due to the migrations, the travels, cases of sleeping sickness in America and other part of the world.
Questions?